Welcome to today's translational research seminar series. I'll begin today's seminar by acknowledging the traditional custodian and owner of the land and on which we meet, and pay my respect to the elders elders past and present. We acknowledge the stories and tradition and living cultures of the Aboriginal and Torres Strait Islander people, and commit to working with them in the spirit of reconciliation, access to justice. So my name is Ren Wang, Senior Research Fellow at Modern Research, UQ. Today is my pleasure to chair the seminar featuring two speakers from AIBN, Professor Alan Rowan and Dr. Amanda Kijas. So they will share their personal insights into the journey of translation. So first up is Professor Rowan. So Professor Rowan is doing his research at the interface of chemistry as well as biology. His pioneer work is processing catalysis and functional self-assembling materials. His research is evidenced by more than 360 publications and with over 20,000 citations. This including some of the very high impact publications in Nature Science, Nature Medicine, Nature Nanotechnology, and he has been awarded 9 patents in the areas of nanomedicine and nanomaterials, and has been involved in the startup of 6 companies. And our second speaker for today's seminar is Dr. Kijas. She is a very passionate research scientist who has been working with Professor Alan since 2017 to develop novel wound management agent and study interface between material and biological process. So welcome, Alan. I guess you to start first.
Want you to start anywhere, or?
I guess okay. It might be easier for you to
move around. Okay, thank you. Well, thank you to try and give you an idea about the journey towards translation. And to be blatantly honest, there is no single pathway of doing it. And I have been involved in setting up 6 companies, and all 6 of them have failed. So that's whether you want to listen to me. You may all want to leave now. And the second thing is, of course, as director of the AIBN, I am regularly involved in the discussions with companies and negotiations and contracts, both the indigenous companies, the federal government, other companies, and see the difficulties it is for an academic to go through that journey. And I think there's a whole pathway of that, which is really, really hard. The only thing I know is that academics aren't great businessmen. And I think you have to accept that. But academics are really smart, passionate people. So being part of the energy and driving a company is really, really important. But I would say try to leave some of the other stuff to the professionals, because ultimately companies only work if they make money, not if they make us happy. Just to clarify that, I think it's a big importance. What do I do in my group? Well, I came out 2017, 22 years in the Netherlands was primarily where I worked. Assistant professor, all through those bits and pieces. And I'm actually a chemist and a biophysical chemist. And I'm going to say from the very beginning, this is a terrible thing to say in this room, don't care about helping people. Right, so let me put that into context, right? I'm going to put myself out there, but I'm really, really passionate about understanding how things work. And the two together is really important. And so I'm not an engineer where I might do formulations. I feel that if you really understand how things work, then you really can get to translation and helping people, because you can build the right thing or design the right thing. So I come from a curiosity driven to do that. And of course, we work in wound materials. We work in understanding how materials work and their interface with biology. We do physics, we do mathematics, we build machines to look at it all, but also ultimately we do want to help people, just to clarify that. And of course, it's never me, it's the people who do all the hard work. So in any of this translation, it's all the people who've worked for me who've done it, not me, right? That's very important. We do things from mechanotransduction, theory tools. We do cells in microgravity, looking at how life would be in space. How can you do scarless wounding? the bleeding problem, which Amanda will look at, and ultimately things like that. And it all started from this. It all started from a strange experiment to try and make wires to go in mobile phones. This wire is 50,000 times smaller than a human hair, and it can conduct electricity, and we were making organic solar cells thinner than a human hair. That's where it started, right? And then we were asked to put it into banknotes. So we worked with the Dutch banknotes to put these polymers into banknotes as a security measure. Abstract and serendipity. And the only way we could get it to work is we had to put them into water so that we could put them in the paper. And then things became strange. And that was the start of the journey. And we can make these polymers here, if this works, which are molecular weight for a biologist. These are 4 million Daltons molecular weight. And they don't wrap up into a ball. They stay as a rigid polymer, and they're made from peptides. And they have unique properties. And that's where we started, by a bit of physics and banknote papers. And loved it. We then jumped into using these polymers for therapeutics, where we would use them to try and control the synapse formation in T cells. And that was my first jump into translation, because we sat down with Kite Pharma and looked at how we could develop this as a new T cell therapy. We were right at the beginning of it. And the first thing I learned here is that the science was telling us we could tune T cells, we could increase memory cells, we could remove toxic memory cells, we could work with the company, but we were a long, long way from a product. And the company was interested with us, and they said, we'll start to have these discussions, but ultimately at the end of the day, their pathway to a commercial product was so far that they withdrew from us. And we put a couple of patents in, and we have some patents on that, and farmers come back to us on it, but it never came to anything because we was too far away from it actually becoming a commercial product. And I want to get that point, is there is a difference between the scope of an academic and translation and working with industry towards a product and actually commercialization. Because when you get into commercialization, and I've been on boards of small companies, it's about, I've been in cases where, what's the packaging going to be like? Who's going to use it? How would the nurse use it? How would you have stability for 5 years? How can you scale it up? What's the GMP? It's an area we don't do, but that's actually what is required to do commercialization. I just want to say there's a difference. We as academics start the pathway of translation. Commercialization is handled by the big boys and money. So we started with that. And then we had a strange observation, and that turned into everything we're going to talk about, is that when you take this gel, because it's stiff and it doesn't form spheres, and you add it to water, it forms a gel. But it does it in the reverse way. As when it's basically cold, it's a liquid. And when you warm it up, it becomes a gel. And that's the reverse you imagine from a gel. It's called an LCST material. And that made it really, really interesting, because we could take this 99.99% water. I could take one bag of it, 1 kilo, and I could gelate a whole swimming pool when the swimming pool warmed up. And that triggered everything on this journey, because this got into the newspapers, this got into Nature, it got in all these accolades. And then all of a sudden the companies came to us because they said, well, we can fill wounds with this and it will set when it warms up to body temperature. Nike wanted to make a bra from it, which stiffened up when you were running. It was used for joints. It was the cosmetics, L'Oreal, all of this thing came up, and that generated interest, which generated opportunity, but also showed how ill-prepared we were for delivering all the samples to these manufacturers and so forth. And we also were hindered by our own success initially. And so that gave us all of that. But one thing this did, which turned it into real science, was the fact that every material in your body, collagen, fibrin, matrigel, has this unique property that when you pull on it, it stiffens up. It's called strain stiffening, not PEG, not other materials, or anything that it wants. And that's a natural aspect of cells and materials. And it turned out we had made a material which had that unique property, and there was nothing else in the world which did it. And so we had novelty. And novelty is really good for translation because nobody else could do it in the world. So that gave us that opportunity to say we have exactly the same thing. We have materials which you pull them and then they suddenly become really stiff. And what I like as a physicist and chemist, I could fit it to an equation, which meant we could control it. So I'm sorry, when I'll go back to it. If you can put an equation to it, you can control it. And if you can control it, you can commercialize it because you know what you're doing. So that's the science side of it, right? So let's jump that. We used it in cells. We take this gel, we put cells in, it thinks it's collagen. Effectively, what happens is you warm the material up, the cells stay there, they grow, they do whatever they want to do, differentiation or whatever. You want to get rid of it, you cool it down, the gel falls away because it's not linked, you extract your cells. And that was the first stage of making a commercial product because we were trying to make two products. One was gels for wound dressings, the second one was gels for cell growth. And so we created a couple, and so we went into this, but I just wanted to come back with one point. I think it's really important. Every single experience I've ever had is that scientific excellence is the key road to translation. What I found is that every time we've raised money, so I will give you a number. I reckon we got $1 million for every Nature, every science paper in translation fund. It came out, it was novel, the companies came to us. I just want to say there isn't a shortcut here. You know, if you know your science, the companies come to you because you're the best at it. They can go and look anywhere in the world, so be very good. And the thing I give to all my academics at the AIBN, where 30 or 40% all funding from industry, don't publish 20 papers, publish 2 very good ones, and you will get more translation. Because companies come to you because you're very, very good at it. They don't come to you as a hobby. And you'll see people around the place who have some money, 30,000 a year or 40,000 over a company. That's not translation. That's just some money generating working with the company. So I just say to you, if you want to get real success and significant money, put your efforts into really good papers. It seems like that you would obviously say that. I know I'm not finished yet, but I will be. So we did. We set up 6 companies. We set up a company which was trying to utilize, and it still does actually exist, looking at these polymers for T-cell therapies, where we would take out the T-cells, we would basically then add our polymers would modify them, you'd get more memory cells and whatever, and that's now being looked at. That is ongoing, but it's only 3 or 4 people. We set up a company for stem cell differentiation and organoid differentiation. Yeah.
People couldn't really hear you.
Oh, sorry.
More close to the mic.
Oh, that's okay. Then I will. Yes, I can't help it. We set up one other. Do we have to carry it around? Look. The circle one. Okay, there we are. Then effectively what we have to do, so we set this up. This worked well with Hans Clevers, of Organoid famous, setting this up. This collapsed at the end simply because we couldn't make enough sterile material. It was purely a commercial entity of how do we scale this up into kilos to get enough sterile material, and it simply cost us too much to do it. But in this journey to set this up, we employed 12 people and we raised 6 million euros, which went back into research into the group. The wound dressings we worked considerably on with the hospital and patients and things like that, and that's something we'll come back to now. That was a grant equivalent to an MRFF of 2 million euros and several smaller grants to try and raise this to develop that. Now, in all of these journeys of setting these companies up, I think the only one which really lasts and survives now is NovioCell. I learned very clearly my role is a bit like in rugby. I get the ball, I have to pass it on. And if I hold on to it too much, I'll never reach the line. And I think there's a really important point here is that what I learned in the first company setting up where I was 5% owner, and I never wanted more than that because I didn't want to be making, I wanted to have some input, but not steering it because that's not what I'm good at, was that my love of science hindered the company because the company simply has to be commercial and get things to market. And it actually didn't care whether it was my polymer or somebody else's polymer. It just needed to get a product. That was the first thing. The second thing is I thought as an academic, I have completely unrealistic expectations of what the pathway is to commercialization. And so through all of these journeys, I've learned basically how little I know. And what I have learned is to hand it over to the professionals at a certain point and be very grateful of that, because they then can run with it. So I can generate the next idea. And that's basically, I think, a message I learned from this. But what I did realize was, though, that there is unlimited funding for that initial stage if you know what, if you have control of your material, you don't have an observation, but you actually have an ability to control it, then companies want to work with you, because they say it's not just an idea, it's really that you have the potential to utilize it. And I think that's something which is quite important to understand. And now, what do we do now? The current work in the Netherlands, of course, that all stopped. We do a lot of this here, is basically we look at osteocytes and osteoblasts, where we use materials to control that. And that leads to a science where effectively we can turn on and off osteoblasts. So the simple lab experiments allow us to turn off osteoblasts to make bone, so osteocytes to control osteoblasts, so that we can understand the drivers of osteoporosis. And in using that science, we can now target areas where we should be delivering new therapies or new diagnostic tools. So this idea of controlling the materials moves on to that. Amanda, you should jump in here, because at this point, all the things I've told you are synthetic. And we came to a point, because this leads straight on to what you want to do in a way, yes, is, and I've just, because we bumped into somebody you'd been working with, who had been using echarine to basically remove platelets from blood. Are you going to mention this?
Yeah, no.
That's how it started. This is a company which has been set up at UQ and at TRI, you know it. It's been around the place, Venomics, QCLA. And that's where it came from, because they'd use snake venom to basically pull out the purified plasma. And they were next door to us. Now, I wanted to use it to control the matrix, because to me, snake venom is just a catalyst, and I can just control the properties of fibrin and therefore control cellular behavior. And then that's where I met this lady, who has a passion for helping people.
Great.
But I don't want you to know. You know what I mean? I'm not allowed to say that. Oh, I did. So, Amanda, you have to help people.
Definitely. And I'm kind of taking over your presentation a bit here, Alan. Okay, he's put in some slides for me, and so that's correct. I have a passion for clinical unmet need and how we can improve, especially in the area of wound management and the area of controlling fibrin networks. And that's a fundamental starting material that after wound is that starting matrix. And those key biophysical properties is how kind of the driver of a lot of our projects, of understanding how that can modulate both cell fate, function, and behaviour. And these are some of Jeff's beautiful fibrin networks using those venom proteins that he can control the architecture of. And that leads into his various projects in modulating fibroblast to myofibroblast differentiation, and that key role in scar modulation. And I'm not going to go into this because I'm still in Alan's talk. Oh, there we go. But and it's a really interesting space that you can, despite the presence of biochemical drivers, that TGF-beta1 that would be usually employed to differentiate fibroblast from myofibroblast, Jeff can modulate his fibrin network architecture to change those outcomes. And that's a space that I'm going to not talk about specifically today, but just highlight those aspects, because it feeds into some of the partnerships that have developed in the recent past. And the key for me as a biologist who used to grow cells on 2D plastic and look at the biochemical triggers of various cellular behaviour was the key role of the biophysical extracellular matrix on modulating cells that can actually almost have a hierarchical control over cell behaviour. And here you can see, because cells aren't static, they continually have this dynamic kind of reciprocity with their surrounding material. And that's a key to a lot of our projects. But the project I want to talk to you about today is more about development and evaluation of these biomaterials in wound management, more specifically and broadly. And even within the space of after injury, transitioning to repair, you had that dynamic change from a fibrin blood clot to restoring that collagen matrix. This completely modulates and changes how cells behave. And we're interested in trying to deliver these for promoting better wound healing outcomes. This has really been driven a lot, and the translational potential with a partnership with Dr. Jason Brown's group at the Hurston Biofabrication Institute. He leads a program in burn surgery, wounds, skin, and his team there. And we're trying to work with him in improving outcomes there in wound management. So the project I'm going to talk about today is actually at the very start of wound healing, which is that formation of that blood clot. And here is probably the closest we are to commercialisation, if you want to call it that. We're still a long way from that. But I want to kind of highlight here is that the project actually started with some really preliminary data. We got some exciting results using these venom proteins. And that was the foundation for actually getting funding from the US military. They were excited by it because it's a new novel approach that hadn't been taken, and we've recently got follow-on funding. So without that, this never went anywhere, and we were quite kind of lucky to get some exciting results that has driven this. The reason why that is because basically in the US military, there's a severe shortage of effective hemostatic agents. Current care, especially in the pre-hospital phase, is tourniquets are highly effective, but these other means to control bleeding aren't. And the later IV-based administration of tranexamic acid, which is a current protocol for how US military combat uncontrolled bleeding, is usually happening quite later in the process. But basically, after major trauma in the US military, the numbers are up to 90% deaths attributable to uncontrolled bleeding. And this is a serious problem because the current agents aren't good enough. So we were looking at this, and we wanted to try and address this using these venom proteins. And a key aspect to this, so these numbers are US military, is that many of these are in the softer tissues in the torso, which are non-compressible injuries, which is the usual route of application, where you have to apply compression with these agents, with these bandages, usually gauze-based, which is, you know, quite old, but still very effective. So basically, this product is that we initiated was these two potent venom proteins recombinantly produced. One, equine, to kind of expedite the blood clotting cascade, the natural endogenous pathway, and that occurs quite late in that cascade, so it's quite resistant to many reasons why other products might fail. And the second component for this topical pre-hospital phase hemostatic agent is antifibrillitic. So this was textilinin, a well-characterised one from Professor Martin Lavin's group from the eastern brown snake, and this stops that blood clot from breaking down again. The key interface here was with these bioactive components that Professor Martin Lavin's group had been working on and using with Professor Alan Rowan's group, where he had this thermoresponsive hydrogel, where this really was amenable for application to wounds that might be quite complex. So to go through that, we did various assays, but the key kind of proof of concept experiments was in a tail amputation model, where we looked at bleeding and how effective this agent, this venom-based hydrogel, would be at controlling bleeding in a normal animal with a functional blood coagulation system. And we saw we really accelerated blood clot formation by 3-fold compared to the natural endogenous pathway, and a 5-fold reduction in blood loss. This didn't require any pressure or compression to achieve this, and When we then looked at an uncontrolled animal, which had been pre-treated with warfarin to disable the blood coagulation pathway, we were still able to really rapidly and effectively control bleeding. And this was a key component because this mimics more where the coagulation pathway might fail. We've also looked at mimics of hypothermia, acidosis, and hyperfibrinolysis, really key coagulopathic kind of base conditions that can occur after trauma. And we did some preliminary kind of systemic immune responses and saw that we weren't seeing any significant response to this product. So that's where that proof of concept kind of product got to, and that's where we're at. So we're really early in the stage, and we're now progressing that to an evaluation of safety and efficacy in a porcine, more high-end traumatic injury model, and looking at how we might develop a delivery applicator, single use by the injured casualty themselves or a first responder. We're looking at how we might ruggedize that for austere kind of function and use, and also alternate delivery scaffolds and other applications. So where you have gastrointestinal bleeding also being a major issue with blood loss. So this has come about through many partnerships, and one of those key ones and why this project was so accelerated was with Professor Martin Lavin and his group in the Venomics area, studying these venom proteins, coming together with the expertise of biomaterials with Alan Rowan. That combination was what kind of accelerated this and really rapidly got some preliminary data that we got really good funding through the US military. Professor Mark Midwinter, ex-trauma surgeon in the Navy, now UQ researcher. He's got the expertise to evaluate these in these high-end pig trauma models, which are kind of a preclinical level. So that's kind of where we also work with the University of Western Australia with 2 researchers there, again in burns wounds. And that's kind of also in a wider space of wound management. Those clinical partnerships, I already mentioned Jason Brown and his group there at the Hurston Biofabrication Institute. This is really fundamental in a broader context for many of our projects, but also in the space of looking at surgical bleeding and how we might be applying it there, and considerations and kind of more clinical considerations of application and ease. And again, Professor Fiona Woods is also a burn surgeon. And I'm mentioning again Professor Mark Midwinter because of his extensive naval trauma experience in that space. A key aspect here is the partnerships that we have in actually kind of progressing this to be a potential product. So as I said, we're really early, but we have these facilities that have been fundamental to progressing this project, from the National Biologics Facility and the Protein Expression Facility based at the AIBN for production of these recombinant proteins and how that might be scalable and usable for a commercial clinical application. As well as, of course, the fundamental role of blood donations from Red Cross has been instrumental in agreements we have with them to be able to access that to look at blood clot kinetics and things. And the commercialisation and regulatory landscape has been 100% feeding in through UniQuest, which supports UQ researchers. And recently they've helped us put together a curator and pulling in Planet Innovation to potentially design that applicator and those key requirements for actual useful delivery, especially in a military kind of setting. So for us, I think many of the key learnings have kind of come down to having really enthusiastic partners that has really helped this along and really driven and, you know, equally passionate to make something happen. And I think something that Alan already mentioned is a bit that we, you know, I'm a very small part, we're a small part of a huge, big partnerships and system, and translation and all that is, you know, so far, you know, such a big kind of picture space that we're a long way from, but we love what we're doing. So the synergistic partners, yeah. So we've got really close synergistic partners that we work really closely with and, you know, work really closely with. We have broader ones and bigger picture ones, and then the commercialisation expertise, which is been going to be really fundamental for these next phases, and the expertise from UniQuest. And maintaining good communication, I think, is maybe a good learning that we've had. Acknowledgements, Alan already started with the group, the broader group, but Rams, a PhD student who's driven a lot of this work along with the rest of the team listed there. Professor Martin Lavin and his team from UQ-CERA has been absolutely fundamental for rapidly getting expertise up and running in the group. Mark Midwinter, Jason Brown, and the various funding, which has, as I said, been really fundamental to even get to this point, and the various facilities.
Can I just say one point? Could you just go back 2 slides, because I think it's a really important point for the translation. One more. So what's really important is this was a strategy from the very beginning to translate this. So the first thing is, no, please stay there. The first thing we did when we did this was we said, if we're going to do this, we have to be able to manufacture it on a proper scale. So we went to the professionals, and we didn't do this in-house. We said, right, make this for us so we can either patent it or we can actually scale it up. So that was the first thing we did. And large enough quantities, we can make this, that's not a problem. So this is now commercially viable, in principle, the mechanisms we have here and here for the 2. The other thing we effectively did was we worked in UniQuest very early from the beginning with regular conversations about how would we develop this. So what we did do is, A, we talked about how we commercialize the different components. B, we talked about on several stages, what are the key experiments we would need to do to get venture capital on board. And C, that thing we were thinking about putting into the soldier, we hire a professional company here to actually go and design it and make it, because we can't do that. And the last thing, which I think is really important, is Amanda now works 60% of her time full-time at Metro North. So you take somebody who was at AIBN and put them over there to make that synergy work, because we could never work it if it was just daily, just talking to people on the phone. So there was a genuine strategy from the beginning to say, hey, we've got something really interesting here, which clots quickly. What is the need, which was quite clear. Well, if we really wanted to translate this, from day one, 3 years ago, when we sent the grant to the US military, we put in place a plan of how we would translate it. We thought about what are the key steps, and we've been working, you'd agree or not?
Yeah. No.
It's not random. There was a real strategy here, working with UniQuest on what would we need to do to get this done in the last 2 years, and also $6 million.
Thank you, Alan and Amanda, for a very informative seminar and presentation. Do we have any questions from the audience? Questions? Okay, I guess we have some time for a couple of quick questions. I'll start. I guess, Alan, you mentioned it's very important to engage with expertise. But can you tell us a bit more how and when did you decide to engage with these industry partners? Because I feel for researchers, sometimes they're a bit hesitate to share ideas.
So the first thing is, I'll be very, I'm very frank. I think people try and hold on to their ideas too long. We get a bit worried about IP ownership and everything like that. I'm always for that, maybe that special knowledge to get it out there and partnership with people as soon as possible for that bit. But I mean, at what point you're toward a commercial partner? I mean, I actually speak to UniQuest often because they've done it. And I've worked with 3 or 4 commercialization units, and I know none of them are perfect, but actually to try and get real advice. Now, there is a lot of funding in Australia for venture capital. I mean, I speak to them and in the Brandon Capital, they say, we've got plenty of money for research. We don't have enough good ideas. And last Friday, I had dinner with the Pfizer, led of Pfizer, and he said, listen, people have to realize what we do as academics is this, and the whole process is that. So you've got to be aware of where you sit in that and what is realistic. And I think there's a much an awareness of us as academics to understand what is genuine translation before we get into that. But I actually would say go and speak to UniQuest, or go and speak to your commercial partner within TRI and say, listen, this is what I think. And actually listen to them when they say, actually, this ain't going to work. Right? Amanda, yeah.
So you've got a lot of partners there, which is great, but also a bit scary. And you said, okay, What is the role of UniQuest in actually telling you to go to AIBN type of innovation? Was that an AIBN?
Amanda?
I think that they didn't play a very big role in those. They were kind of earlier partnerships. UniQuest is now coming in more strongly now, I feel, and like with this curator that we put in with Planet Innovation to try and promote that applicated device. So this is, you know, still at early stage, but oh, sorry. Yeah, so it is many partners, but you know, some of them are facilities, some of them, you know, they're all feeding in different ways. And I think, you know, we can't do everything. I mean, we had really interesting results that, you know, went amazingly, you know, and we were excited by them.
How do you figure out the Planet Innovation even?
That came from UniQuest.
That was UniQuest, actually, yeah. So, and actually the Australian Defence Force people we were speaking to, they spoke. So I shouldn't be promoting a company, but that was, I mean, it's only the applicated device, and they're in that space of healthcare innovation and things. And yeah, so it was a recommendations from Australian Defence Force, which we've only kind of more recently engaged with, and then UniQuest also.
But I do think you have to take that professional step one step further. So, you know, talking to the Australian Defence Force, even to the point where we said, you know, we're thinking of building this, do you think that soldiers will use it? Is actually quite useful. And I think that will, by having those conversations and being open, there is more chance of this getting better. The other thing is we are not wed to anything. We talk about the gel we use now. I don't care what gel I use. I don't care whether I use textin or a protein. But we've got a mechanism which works really well. How can we get that optimum to eventually help? And I'm being convinced help.
Yeah, you're helping
patients.
I think we also have questions on the chat. If we could have a look at I can. Yeah, so people are interested to know about a large amounts of money available for research.
So
US military have a lot of money if you're competitive enough to get it.
So it's a difficult one. I mean, money, yeah, there is. So I work with Random Capital, and they say there is venture capital. And my experience is there is money here, but it's not always easy to get it out of them. Because I will argue and say also that Australia is quite risk averse compared to Samoa, so it is harder. And so I do think that the opportunities in other places where you could go in and say, hey, I've got this, it's interesting, which might resonate, say in California or Singapore, doesn't happen here. So when you do go into those, you've got to be far more savvy about the commercialization pathway. So that's why I would have that conversation with your commercialization person in your building, or I would have the conversation with UniQuest before you go and talk to the company, so that you're a little bit more switched on, because they're not quite so easy to give up free money. But I mean, when I speak to the ministers, whatever, there is money there. We've got 20 billion in MRFF and things like that. You've just got to be a little bit more commercial savvy and put things in place before you're going to raise it. I'm not sure if that answers the question. But I just think it's not like it's in other places where I've got a smart idea and I get funded. It doesn't work like that in Australia. That'll be more organised, yeah?
Yeah. And I guess we have a final question from Michael Roberts online. He's asking, can you comment on the viability of your various markets? Is there a threshold you have to meet for a given strategy to be worthwhile?
Well, if you talk to UniQuest, it's about how much does each one cost. If you talk to the military, if they can save a large percentage of their soldiers, then it's priceless. We're continuing the conversations because there is nothing else on the market to save these people. And our goal is to actually probably have this in every market. So that basically, if you've got uncontrollable bleeding or whatever, you just jab it in, it seals in 30 seconds, and that gives you enough time to go to the hospital. So there is a novelty factor of it. There is a huge market. I mean, the market is 6 to 10 billion just on the US military side, in that sort of level. And that's attracted the interest of the more venture capital, because they see there's an end user who has an interest in buying it. So in his question, the viability, I would say it's probably 75%. In my experience, this one will fly.
Yeah, awesome. I guess if there is, okay, we have one last question from Nick.
What happens with regards to thrombosis or heart attacks? If you have the neck heart
Yes, so currently the eccrine procoagulant is covalently bound to the polymer, so it's localized to the wound.
Yes.
So that's the current safety thing. But actually, we haven't done animal-based evaluations where we actually don't think we do need to do that because it's so potent at clotting, it doesn't move, it doesn't get a chance to move. But at the moment, it's covalently bound to the polymer, so you know, in a way that commercially not as viable approach because it's more processing, but we actually think just co-delivery of a gel with the venom proteins would be enough. But yeah, that was the safety issue because it is potent. I mean, it's nanomolar amounts of this is clotting.
So I mean, that's the issue with all of this is the FDA approval on the inside the whole pathway. And hence, to be honest, in the question of viability, any medicine is more medical thing, there is that risk. And so how do we mitigate that risk? We're mitigating that risk by actually having the US military interested in buying our products. And that's probably when I say it'll get through, because the venture capital people might not take that risk if they didn't see a clear market at the end. Yeah,
so they're funding now, just this year, the preclinical kind of evaluation, these iron porcine, benchmark models for hemostatic agents. So we're kind of pushing to that next level. So, we're still early, but yeah, it's really promising that they're still supporting that, with that proof of concept kind of data that we have.
Thank you very much for a very interactive and informative session. Thank you. I guess with that, we'll conclude today's seminar.